Eosinophilic esophagitis, or EoE, is a chronic allergic disease associated with type 2 inflammation and epithelial barrier dysfunction. The etiology is unknown. However, genetic heritability studies suggest environmental factors play a key role in pathogenesis. Detergents like SDS or SLS are common ingredients in household products like dish soap and toothpaste and have been shown to disrupt airway and skin epithelial barriers. We hypothesize detergent exposure decreases esophageal epithelial barrier function and induces esophageal inflammation. To explore this hypothesis, we utilized in vitro and in vivo approaches. We found evidence of disrupted barrier function and increased inflammation. We propose that these responses may be occurring in humans with exposure to common household detergents such as SDS and toothpaste, and this may contribute to EoE pathogenesis. Using air-liquid interface cultures of human esophageal epithelium cell line EPC2 exposed to concentrations of SDS exposure roughly 1/6000th that found in toothpaste, we showed decreased electrical resistance in panel A and increased permeability to phytidextrin in panel B. At this concentration, we also found increased expression of the pro-inflammatory cytokine IL-33 and decreased expression of epithelial junction molecule desmoglein-1. Next, we exposed mouse esophageal epithelial organoids to SDS and showed disrupted layers of undifferentiated basal cells and irregular tissue matrix. Next, we administered 0.5% SDS in drinking water to mice for 2 weeks. We found striking esophageal inflammation, including eosinophilia, highlighted here by EPX staining. We also found increased CD4, suggesting the possibility of an adaptive T-cell mediated response. Here we show a close-up of an eosinophilic microabscess in a mouse from the SDS treatment group. Also of note is the basal zone hyperplasia and spongiosis indicated by yellow arrows in the middle panel. Finally, basal zone hyperplasia was also observed by Ki67 staining, and we found increased IL-33 staining. Together, these changes are consistent with observations in humans with EoE. Alarmingly, we found that in 1939, when soap was being replaced with synthetic surfactants like SDS, these authors found that oral exposure in rats to low concentration SDS induced gastrointestinal irritation, especially of the esophagus. We echo their concerns and suggest more research is needed to understand the potential contribution of common household detergents not only to EoE but a host of diseases. We gratefully acknowledge the generous support that made these studies possible. Please look us up on Twitter at Doyle Lab.